from the Consiglio Nazionale delle Ricerche in Italy, and Professor Mancarone is from University of L'Aquila, also in Italy. Marzia Arese from Sapienza University of Rome, Elena Forte from Sapienza also, and Alessandro Leuti belonging to the Campus Biomedico University of Rome. In particular, this webinar was organized in cooperation with the Coast Action CA 20121 that is coordinated by Professor Antonio Quadrado. And the name of this action is From Bench to Bedside Transition for Pharmacological Regulation of NRF2 in Non-Communicable Diseases. And I leave the floor to Antonio for the introduction of the first speaker. You are on mute, Antonio.
So thank you very much, Luciano. It is a great pleasure to be here again with this seminar series that we share with the Sapienza University. And I'm going to start just by introducing the first speaker of today, who is Dr. Elke Hayes. She is Associate Professor of Pharmacological Biology at the Department of Pharmaceutical Sciences at the University of Vienna in Austria. She is interested in signaling pathways involved in the cellular stress resistance and their rational modulation by small molecules. For the past years, her main research activities have been focused on dissecting the crosstalk between cellular redox and energy stress in the molecular level, and on the identification and characterization of novel small chemical entities with benefit for the cell's capacity to efficiently cope with stress. Thank you very much, Elke. The floor is yours.
Yeah, thank you, Antonio, for this kind introduction, and thanks to the organizers for having me in this distinguished webinar series. It's really an honor to be one of the speakers. So, and of course, thank you to all participants for being with me in this presentation. So let me just go back. No, I don't. Does this work? Okay, so here we go. Do you see my screen?
Yes.
Okay, wonderful. So welcome to my talk, and as already mentioned, I will guide you through a little bit of signaling crosstalk between NRF2 and the metabolic regulator AMPK. So actually, before I go into our data, I would like to give you a short introduction of the two players, NRF2 and AMPK, although I do think that NRF2 doesn't need a lot of introduction in this kind of audience. But still, to bring us on the same page, NRF2 is a transcription factor. It stands for nuclear factor E2 related factor 2. As already said, it's a transcription factor, and it's a stress responsive transcription factor. That means that under basal conditions, NRF2 abundance is very low in the cell. So, but upon exposure to different stressors, and these can be oxidative stressors, xenobiotic stressors, but also metabolic stressors, NRF2 gets stabilized. It's not any longer degraded by its inhibitors, and it moves into the nucleus where it can bind to its DNA consensus sequence, the so-called antioxidant response element, and it can trigger target gene expression. And the target genes of NRF2 are manifold. There are around more than 250 ARE-dependent target genes of NRF2, and they actually belong to proteins that sort of detoxify the initial insult which was triggering NRF2 activation. So they belong to the proteins injuring redox balance, but also metabolic genes are expressed upon NRF2 activation, and anti-inflammatory proteins are also involved in the NRF2 signaling pathway. When we look how NRF2 is built up, we can see that it has different domains, and as it is a transcription factor, it has a DNA binding domain and a transactivation domain. But it also has other domains which are important to interact with the inhibitors of NRF2. So interaction of the NEH2 domain with KEAP1 leads to ubiquitination of the transcription factor and to its degradation. And the same is true when the NEH6 domain is interacting with beta-TRCP. But apart from regulating the abundance by degradation, NRF2 can also be regulated in terms of abundance by regulation of synthesis. And it also can be post-translationally modified either by phosphorylation, acetylation, or sumurylation. And there are some other post-translational modifications coming up soon, I guess. And these post-translational modifications finally fine-tune activity and localization of the transcription factor. So this already brings me to the second layer of my talk, and this is AMP-activated kinase, or briefly AMPK. It's a serine threonine kinase, which comes along as a trimer. It's built off of an alpha catalytic subunit and two regulatory subunits, the beta and the gamma subunit. The regulation of AMPK is mainly on a post-translational modification level or occurring on an allosteric level. An allosteric activator of AMPK is, as the name might suggest, is AMP, or anything which leads to an increased ratio of AMP over ATP in the cell. Phosphorylation and activation of the kinase is mediated by upstream kinases, and there's all the TGF beta activated kinase, the calcium calmodulin dependent kinase, kinase 2, and the liver kinase B1. And there's another site in the beta subunit of the AMPK, which is called the ADAM site, and this stands for allosteric drug and metabolic metabolite binding site, and binding of endogenous or exogenous molecules can also lead to activation of AMPK. But what happens upon activation of AMPK? AMPK is phosphorylating a large range of substrates. And once these guys are phosphorylated, they sort of shut down ATP consumption and trigger ATP production. So in this sense, AMPK, an activated AMPK can increase glucose uptake, glycolysis, mitochondrial synthesis, and it can shut down synthesis of fatty acids or cholesterol. But AMPK is also a redox sensitive kinase. It can be activated by redox stress. This happens by modification of the alpha subunit or by activation of these upstream kinases. And it can also trigger antioxidant responses. This is very crucial for the vasculature, where AMPK phosphorylates eNOS and then leads to increased NO production. AMPK can also trigger autophagy. That's also important to keeping up the metabolic homeostasis, but it's also important in terms of cleansing the cell from rubbish. So AMPK is triggering autophagy. Okay, with these two players introduced, when we look at them, we kind of see that the same insults and also an overlapping set of small bioactive molecules are often found to activate both signaling pathways. And once activated, each of these signaling pathways elicit similar downstream phenotypic responses. So we have seen that energy stress, redox stress, xenobiotics, or proteotoxic stress, which all lead to a disruption of the cellular homeostasis, they can act positively on the activity of the AMPK and/or NRF2 signaling. And once these tools are activated, it comes to a restoration of the cellular homeostasis, if you will so. So, and then now the question which actually occupied us for a long time was, do they sort of know from each other in the cell, or do they just act in parallel in a blind fashion, so to say? Nobody knows from the other, or do they really crosstalk with each other? And actually, this question not only occupied us, but a lot of other people in the research out there, because I checked today PubMed. So if you put an AMPK and NRF2, there are over 680 scores in PubMed. So, and apparently there seems to be a direct crosstalk between NRF2 and AMPK. So, and in the next slide, I just want to briefly guide you through the data which show that there is definitely a crosstalk between AMPK and NRF2 and how this is realized on the molecular level. Of course, a very obvious thing how AMPK could interact with NRF2 is by phosphorylation, so that the kinase phosphorylates the transcription factor. And this was already shown in an in vitro kinase assay where AMPK and NRF2 were put together, and a phosphosite on NRF2 was identified, which was a serine 550 in the murine or the serine 558 in the human sequence of NRF2. When this phosphorylation site is mutated, NRF2 shows a reduced nuclear localization. So AMPK boosts the nuclear localization of NRF2. Another way how AMPK can influence NRF2 signaling is by its ability to inhibit GSK3 beta activity. And GSK3 in turn inhibits NRF2 signaling, and it does so by priming it for beta-TRCP mediated degradation or by triggering nuclear exclusion. So activated AMPK by inhibiting GSK3 beta can have a positive influence on the NRF2 signaling pathway. Another way how AMPK could sort of merge into the NRF2 signaling is via its activating potential of autophagy. So as seen in one of the previous slides, AMPK can trigger autophagy, and it does so by inhibiting mTOR signaling or activating the ALK kinase, which is a very important kinase in the autophagy machinery. And by phosphorylating P62, AMPK can sort of increase the affinity of P62 for KEAP1 and tag it for degradation, if you will, so by autophagy. So by activating autophagy on the one hand side and by increasing the affinity of P62 for KEAP1, AMPK leads to the autophagic degradation of KEAP1, and KEAP1 being a negative regulator of NRF2, AMPK activates NRF2 in this pathway. Another possibility is that AMPK not only phosphorylates NRF2 itself, but it can also phosphorylate histones or other epigenetic regulators. And AMPK, as being one of the prime regulators of metabolic pathways in our cells, it also sits in the control room over the metabolite levels in our cells. And you might know that the metabolites in our cell, like acetyl-CoA or S-adenosylmethionine, are not only building blocks for other bigger molecules in our cell, but they are also needed for post-translational modification on proteins or on epigenetic regulators. Though this influence of AMPK on the chromatin arrangement is very well known, and of course, it affects gene expression, but the direct influence of the epigenetic influence of AMPK on NRF2 hasn't been shown or been studied to a great extent yet. Another way how AMPK could signal to NRF2 is in the field of the retrograde mitochondrial nuclear signaling pathway. This MODS-C, what you see here, it's a mitokine, meaning it's a mitochondrially encoded small peptide. And mitochondria produce these peptides upon AMPK activation. And this small peptide then moves into the nucleus, where it was shown to be bound on NRF2, sitting on ARE consensus sequences. And by this binding, it leads to a stronger transactivation of NRF2 target genes. So this could also be a way how AMPK could positively influence NRF2 signaling. So these were actually the ways which have been reported, or the theories where we think that AMPK might influence NRF2 signaling. And we also digged into the influence of AMPK on NRF2 dependent gene expression, because mainly we were coming from the field of bioactive natural products. And when you ever have a beneficial natural product at hand, the activation of NRF2 and AMPK is not really far away. So whenever you check what bioactivity does this natural product have, NRF2 and AMPK is for sure popping up at some point. So we were wondering, what is going on between these two. So we wanted to add something onto this picture. In a very first step, we took a rather simple assay. So we took mouse embryonic fibroblasts, which were wild-type and AMPK knockout or NRF2 knockout cells. We treated them with an NRF2 activator, which also activated AMPK signaling, in this case sulforaphane, and assessed the gene expression profile by microarray. And then we performed STAT analysis and pathway analysis. And what we could see that from the genes that were NRF2 dependent, only a subset of these genes was also AMPK dependent. So AMPK seemed to only affect a subset of the NRF2 regulated genes. And when we digged into this microarray data a little bit deeper and also tried to validate this data by qPCR, we saw that genes belonging to the glutathione metabolism, drug resistance, or chemokine signaling were rather not dependent on AMPK. So we could see even induction in the wild type and the AMPK knockout cells, whereas for other genes, AMPK presence really mattered. And these genes belong to the axon guidance, focal adhesion, PX3 kinase signaling, or the pluripotency signaling pathway, and also heme oxygenase show this particular phenotype that in wild-type cells, it got much higher expressed than in the AMPK knockout cells. So then we wanted to know what is the reason beyond this, and we checked all the theories which I have explained to you in the previous slides. So we didn't see any difference in the nuclear translocation of NRF2 between wild-type and AMPK knockout cells, and we didn't see any difference in the KEEP NRF2 interaction between wild-type and AMPK knockout cells. So AMPK did not influence the nuclear translocation of NRF2, nor did it influence the NRF2 KEEP1 interaction. Also, when checking the chromatin accessibility of the ARE sites of the investigated genes, we could not see a major difference, or when we saw differences, they occurred rather isolated and did not really match our observations in the microarray analysis. So overall, there was no general or consistent influence of AMPK on the chromatin opening around the ARE sites. But compelled by a study which was ongoing in our team at the same time, we were compelled to look at BAH1 levels. So BAH1 actually is a protein that also binds to the ARE sites of the DNA, and it competes for these sites with NRF2, but BAH1 usually acts as a repressor of gene expression. So we were wondering, maybe this could be an explanation for our observation. And actually, we could indeed see that the AMPK knockout cells showed higher levels of BAH1 compared to the wild-type cells. And this also was true for the nuclear levels of BAH1. So when we compared the NRF2 BAH1 ratio in the cells, we saw much higher NRF2 over BAH1 in wild-type versus AMPK knockout cells. And just to see whether this is not an artifact of our knockout cells, we also confirmed the negative impact of AMPK on BAH1 via siRNA experiments. We also could find out that the BAH1 is rather regulated by AMPK on the de novo synthesis level. So the mRNA of BAH1 seemed to be sort of downregulated by AMPK activity. This led us then back to our observation of the gene expression analysis. So we're wondering, okay, maybe at the ARE sites in the wild-type cells, there's more NRF2 than BAH1 than in the AMPK knockout cells. And this is actually what we also saw. So if you see, so these are the ARE sites of two selected genes. And we do see that in the wild-type cells, we do see higher NRF2 levels over BAH1 levels. Whereas in the knockout cells, we do see pretty much we don't see such an enrichment of NRF2 over BAH1. This might very well explain why why AMPK can boost the NRF2 signaling, but it didn't explain why some genes are AMPK responsive whereas others are not, because this enrichment of NRF2 over BAH1 in wild-type cells occurred in AMPK responsive as well as in non-responsive genes. So this couldn't be the sole explanation for it. So then we looked for another explanation, and we went back to the post-translational modification issue. What we did is that we transfected a fusion protein of NRF2 in different cells, like HEX cells and wild-type and the knockout cells, and treated them with solvent or AMPK activator, and did a mass analysis of the immunoprecipitation. And what we came up with are 3 phosphorylation sites, which are phosphorylated in an AMPK-dependent manner in living cells. And we also could confirm that these 3 serines are AMPK substrates in vitro. So doing an in vitro kinase assay with just AMPK and NRF2, putting it together, we saw that these 3 sites really are phosphorylated by AMPK, whereas an unaffected phosphorylation site was not. Okay, so this didn't, and then we wanted to see whether AMPK is also the direct kinase which phosphorylates NRF2. For this, we still don't have an absolute proof, but we do see interaction between NRF2 and AMPK in cells by co-immunoprecipitation experiment and also by proximity ligation assays. Then we wanted to check what these phosphosites do with NRF2. We mutated these 3 phosphosites, and this is called the triple mutant of NRF2, where the serine residues are mutated to alanines. And if we co-transfect these two forms of NRF2, like the wild-type and the triple mutant, into the cells and check the decay of NRF2, we do see that there's no difference between wild-type and the triple mutant. And this is true for 2 different cell lines where we introduced these versions of NRF2. So the phosphosites do not alter the NRF2 half-life, and the phosphosites also do not alter the nuclear translocation. So we definitely see the wild-type is translocated to the nucleus up until forfein treatment, and also the triple mutant is translocated to the nucleus. And this is also shown by confocal microscopy. And also, if we look on the transactivation potential of a reported gene assay, we see that the wild type does pretty much the same job as a triple mutant. So general transactivation is not impaired by mutation of these phosphorylation sites. But then we went back to our AMPK responsive and non-responsive genes. What we did is that in an NRF2 knockout background, we transfected the mutant forms and the wild-type form of NRF2 and checked what is happening with the target gene expression up until forfein treatment. And there actually we could see the difference. Whereas the GCLC, and remember, this was an AMPK non-responsive gene, we don't see any difference between wild-type and triple mutant. But looking at hemooxygenase, which was an AMPK-dependent gene, we could clearly see that with the wild-type NRF2, it experiences a much better induction than by the triple mutant of NRF2. So the phosphosites seem to really affect the extent of target gene expression in a gene-selective manner. And this nicely mirrors the picture we have seen in the wild-type versus the AMPK knockout cells. So this already brings me to the summary where we are now at the moment, showing that AMPK presence apparently goes along with a changed amplitude in expression of a subset of NRF2 target genes. And AMPK also leads to a reduced level of BAH1. And with these reduced levels of BAH1, AMPK leads to a tilted ratio of NRF2 versus BAH1 bound to the ARE promoter and enhancer sites, but it cannot really generally explain this gene selective influence we have seen in the microarray. For this, we rather think the phosphorylations at serine 374, 408, and 433 come into play. And because we have seen that these phosphosites are phosphorylated in an AMPK-dependent manner, and mutation of these serines causes the selective reduction of the NRF2 target gene expression, which nicely mimics our picture seen in the comparison between wild type versus AMPK knockout cells. But still, there's plenty of work to do because we still don't quite understand how these phosphorylation sites really affect the selective impact on the NRF2 target gene expression. Is it a general theme for all the AMPK responsive genes, or is it really a gene-specific effect? We also need to find out what the role of these phosphosites is in terms of promoter complex composition or the ARE binding behavior of endogenous levels of NRF2, and also if there is a cooperativity and a functional crosstalk between the observed AMPK BAF1 and the AMPK phosphor-NRF2 axis. And of course, we work rather with cell culture, with in vitro systems, which definitely are quite far away from the physiological situation, which might really have a lot of artifacts and pitfalls. So we really need to also show and find out how our findings are really contributing to the stress resilience capacity, which is definitely found to be increased during caloric restriction or by exercise, which actually leads to AMPK activation. So I think there is a crosstalk also in the physiological situation between AMPK and NRF2, but still we need to dig into whether this is phosphorylation site dependent and understand how these might happen. So with this, I'm pretty much in the end of my talk, and I wouldn't like to end without thanking the people involved in this group, in this work. So this is our group at the University of Vienna and my cellular stress resistance team. And the work I presented was mainly initiated by Manuel and Katrin and is now continued by Eleni and Sarah, and hopefully with as nice results as the other two. Our collaboration partners and the funding should also be acknowledged. And I thank you very much for your kind attention. And I'm happy to take any questions either in the chat or later on after the other two speakers have delivered their talks. Thank you so much.
So thank you very much, Elke, for a very interesting talk. I repeat to all participants that we are not going to open the microphones, but we ask all of you to put the questions in the chat because we will have the discussion at the end after the three talks. So now we proceed to the second speaker, and it is a pleasure to give the floor to Gerasimos Sikiotis. He will give a talk on translational implications of NRF2 signaling in the thyroid. He is an associate professor and a senior staff physician in the University of Lausanne, and particularly at the Faculty of Biology and Medicine of the University of Lausanne, and a senior staff physician at the Service of Endocrinology, Diabetology and Metabolism of the Lausanne University Hospital. Jerry is a physician scientist specialized in clinical and basic endocrinology, with a particular focus on thyroid physiology and thyroid diseases, including hyperthyroidism, hypothyroidism, and thyroid cancer. Since 2015, he is responsible for the thyroid endocrine clinic at Lausanne University Hospital. His clinical research focuses on the needs of patients with thyroid diseases, including quality of life among thyroid cancer patients and survivors. His basic research, funded primarily by the Swiss National Science Foundation, focuses on the roles of cellular homeostatic systems in thyroid physiology and pathophysiology. So with this, it's a pleasure to give you the floor, Jerry, and thank you for participating.
Thank you very much, Luciano, for the invitation, also to the other organizers and the Benefit for Action, and for the presentation, the introduction, I mean. So we will discuss NRF2 signaling in the thyroid gland, and we will touch a little bit on what this means for metabolism. For those who are familiar with the thyroid, I think everybody's familiar with the location of the thyroid in the lower neck, in front of the trachea and the larynx. But most people may not know that the thyroid is actually composed of these little spheres. They are called thyroid follicles. So the epithelial cells are in the periphery of the sphere, and the synthesis of thyroid hormones takes place on the surface of the cells towards the center of the sphere. So this is what we call the apical surface. We will discuss how the synthesis takes place in more detail in the next slides. Thyroid hormones act on all cells of the body. They influence many processes, including differentiation, growth, and of course metabolism. Specifically about metabolism, thyroid hormones determine the basal metabolic rate, and they do so by stimulating brown adipose tissue. They stimulate the sympathetic nervous system, which innervates brown adipose tissue and increases metabolism by increasing the mitochondrial activity. Thyroid hormones are then required for the homeostasis of all tissues. Here we see in some more detail the synthesis pathway or mechanism of thyroid hormones. Sorry, I don't know how to get rid of this bar on the top from Zoom. I don't know if you can see it and if it's bothering you. Yes, maybe.
We don't see the bar, so it's perfect.
Okay, great, thank you. So thyroid hormone synthesis involves oxidation. And here you have a slice through a thyroid follicle. On this side, we have a blood vessel. This is an epithelial cell. And here is the center of the follicle. It's what we call the colloid. It's a viscous substance. So basically, in order for the thyroid to synthesize its hormones, it needs two basic components. One is iodine that comes from the diet, and it enters the thyroid follicular cells through a dedicated transporter. It's called the sodium iodide importer, or NIS for short. And the second component that is needed are tyrosines. These tyrosines, they come from a protein that the thyroid cells synthesize. It is called thyroglobulin. This is the precursor of thyroid hormones. And it has specific tyrosines at which thyroid hormone synthesis takes place. So thyroglobulin is synthesized, and it is also, together with iodine, both of them are moved from the interior of the cell to the exterior of the cell, meaning in the colloid, here close to the apical surface. And this is where thyroid hormone synthesis, or most of thyroid hormone synthesis, happens. It happens through a series of oxidation reactions. The first one oxidizes the iodide anion to iodine. So it changes the molecular form or atomic form of iodine. And then this form of iodine, through another oxidation reaction, can iodinate specific tyrosine residues. And you end up with tyrosine residues on thyroglobulin that have either two iodine atoms or one iodine atom. And then in the next oxidative reaction, these iodinated, either monoiodinated or diiodinated tyrosines, they are conjugated. And they form these molecules either with 4 atoms of iodine per molecule of thyroid hormone attached to thyroglobulin. This is levothyroxine, or T4, or a molecule with 3 iodine atoms in these specific positions on the thyroglobulin backbone, and this is T3, or liiothyronine is the chemical name. Basically, we'll just call them T4 and T3 for simplicity, and it reflects the number of iodines per molecule of hormone. In this case, the hormones are still attached to thyroglobulin, so then this iodinated thyroglobulin is taken up by the cell by endocytosis. These vesicles, they fuse with lysosome. The iodinated thyroglobulin is degraded, hydrolyzed, and then T4 and T3 are released on the other side. These vesicles, they fuse with the membrane, and they release thyroid hormones in the extrastitial space and eventually back into the circulation. If we summarize this a little simplistically, we can say that in order to make thyroid hormones, start with iodine, and we need oxidation reactions. And these oxidation reactions use hydrogen peroxide. So the thyroid epithelial cells have dedicated enzymes, they're called dual oxidases, that actively generate hydrogen peroxide. And there is an enzyme on the apical membrane here that is called thyroid peroxidase, and it catalyzes these three oxidative reactions that are necessary to iodinate thyroglobulin. And in the rest of the process, the iodinated thyroglobulin is taken up by the cell, and T3 and T4 are liberated in the bloodstream. So, since thyroid physiology requires oxidative reactions, it is not surprising that oxidative stress or a certain level of oxidative stress is important for thyroid function and homeostasis. And it has been shown, for example, that, again, a certain level of oxidative stress is required for normal proliferation and function of thyroid cells. And not surprisingly, then, the thyroid also has increased tolerance for oxidative stress. For example, the spontaneous mutation rate in thyroid cells is lower than in other tissues, and the levels of oxidized DNA are lower as well. In certain conditions where there is even more oxidative burden for the thyroid needs to react. This happens, for example, when there is not enough iodine, or under conditions that lead to enlargement of the thyroid, what we call a goiter. Those conditions, we move away from what is considered physiological stress, or some people might call eustress, and we move into the area of pathological oxidative stress, in which case the thyroid needs to react by an antioxidant and detoxification response. So this was all known before we started our work in around 2010. And at that time, we had the following questions. We wanted to know how thyroid follicular cells sense oxidative stress, how they launch these antioxidant transcriptional responses, and specifically also how the redox status of the thyrocyte is coupled to iodine metabolism and the hormone synthesis machinery. And we speculated that the NRF2 system, because it is very important in antioxidant defense, and because in every tissue where it has been studied, it has been shown to have some tissue-specific functions as well, we thought it may have something to do with the physiology of the thyroid in relation to these questions. One experiment one can do to generate oxidative stress in the thyroid is to give mice additional iodine. So I mentioned before, reducing the iodine in the diet causes oxidative stress, but giving an overload of iodine at pharmacological doses can also cause an antioxidant response. So in this work, we characterize this antioxidant response in the transcriptomic response of the mouse thyroid to iodine overload, and we did this in wild-type mice and in NRF2 knockout mice. This was the experimental design. So you have your mice under normal conditions, or to those mice where we gave excess iodine, it is easy to do technically. You dilute a high concentration of sodium iodide in the water that the mice drink. And after 7 days on this condition, the mice are sacrificed. We remove the thyroid, we take the serum, we harvest some other tissues, and we analyze them. In this case, we did RNA sequencing on the thyroid glands, so we could see genes that upregulated in response to iodine, or downregulated here, by comparing wild-type mice treated with exposed iodine to wild-type control mice, and so on for the other groups and conditions. What you can see is that one of the pathways that were significantly changed in response to iodine in an NRF2, was the NRF2 response, because the response of all these genes was dependent on the NRF2 genotype. These results came from the analysis of the data using the ingenuity pathway analysis. In what I showed you before, each column was pooled data from the mice. Here, each column is a single mouse of the respective group. And when we looked more specifically at the various cellular processes that were differentially affected in the different groups, we could conclude that NRF2 has a protective role against the inflammatory, immune, and fibrosis response that is triggered by excess iodine. We looked at some antioxidant target genes in more detail. Here you can just see the NRF2 levels, showing that there's no basic low expression in the mice. And we looked at NQ1, the typical prototypical NRF2 target gene, which is highly upregulated in response to iodine in wild-type mice. Its basal levels are reduced in knockout mice compared to controls, and there is no inducibility. And other antioxidant genes, glutamine peroxidase 2, thyroxine reductase 1, behaved in more or less similar manners. The enzymatic activities of glutamine peroxidase 2, glutamine peroxidase, and thyroxine reductase have been known for decades to be important in thyroid physiology, in the normal function of thyroid follicular cells. Here we looked at the levels of oxidation of proteins in the thyroids of these mice. Here again, each column represents pooled samples from several mice. And you can see that the only group, this is the oxyblot method, and it detects oxidized proteins. You can see that the only group that stands out is the knockout group treated with iodine. There are no statistics on this graph because it is an N of 1, but I think you can appreciate the difference. Also, when we looked at the levels of oxidized lipids in the form of 4-hydroxynonenal, we could see the same. So here you have follicles of a wild-type mouse with no iodine, and around them you have fat. So this serves as a positive control staining here. And again, you can see that the only group where the follicles are stained is the knockout group after treatment with iodine. We conclude from this data that NRF2 protects against excessive oxidation of proteins and excessive oxidation of lipids in response to treatment with iodine. This is not very surprising because we know that this is what NRF2 does, it protects against oxidative stress. What was surprising is that we found that NRF2 also regulates directly the production of thyroglobulin and its iodination in the thyroid. So it also has something to do with the thyroid hormone synthesis machinery. These are the mRNA levels of thyroglobulin in the different groups. You can see that there is no impact of iodine treatment, but the levels are much lower in the knockout mice. You could see the same at the protein level. Here, each column is thyroid protein from a single mouse. And already on the Kumasi stain gel, we can see thyroglobulin bands up here. And we can see that they are fainter in the knockout mice. And when we do Western blot for thyroglobulin with a specific antibody, we can see that its levels are decreased in the knockout mice. We observe the same pattern in the wild-type mice, in the mice treated with iodine. So the difference is independent of iodine treatment, and the knockout mice have lesser globulin, mRNA, and protein than the wild-type mice. In order to study this in more detail mechanistically, we took an established cell line of rat thyroid follicular cells, and we knocked out NRF2 or KEAP1 by CRISPR-Cas9. So we made mutant cells, homozygous mutant cells, and we use them in some of the experiments I will show you here. In those cells, we could replicate everything I showed you so far, meaning the transcriptional response to iodine, its dependence on NRF2, the lower mRNA and protein levels of globulin. So these cells replicated the in vivo phenotypes very well, so we could use them further mechanistically. The enhancer, the regulatory area of globulin, we saw that it contained two potential binding sites for NRF2. You can see them here in green. What you see in red and in yellow are binding sites for other transcription factors that have been shown or postulated to regulate the activity, the transcriptional activity of the thyroglobulin gene. You can see actually that the AREs are much better preserved between humans and rodents than most of the other binding sites. So we cloned this upstream enhancer in a reporter construct, and we transfected the thyroid cell line I mentioned, not the one with mutant NRF2, but with wild-type NRF2. And you could see that in those cells we can stimulate the activity of the reporter by treating with sulforaphane, an NRF2 activator. When we mutagenized each one of the AREs, the basal activity was reduced and the inducible activity was also reduced. And when we mutagenized both of them to sequences that could not bind NRF2, the basal activity was reduced further and there was no inducibility, which indicates that both of them are functional. By doing chromatin immunoprecipitation experiments in cells, we could show that NRF2 binds to both of these AREs. As a control, we use the MK1 ARE. So basically, NRF2 directly controls, as a conclusion, directly controls TG gene expression by binding to two functional AREs in a conserved upstream enhancer of the gene. Then, what was surprising to us and in a disappointing way, to say, was that these mice did not have a difference in thyroid function. Even though they had then less thyroglobulin, they managed to generate the same amount of thyroid hormone. What we use to assess thyroid hormones, both in mice and in humans, the most sensitive marker is the the pituitary hormone that controls the function of the thyroid. So this hormone, it's called thyroid stimulating hormone. It acts on the epithelial thyroid cells to stimulate their growth and their function, and all the steps in thyroid hormone synthesis are stimulated by TSH. So it is very sensitive to even small differences in the levels of T4 and T3. This is why it is the best marker that we use. And you can see that there were no differences in the serum levels of TSH between the two genotypes. And in the iodine-treated mice, there were no differences either. The T3 levels in the serum were unchanged between the genotypes, and there were differences in the total T4 levels. We could see these differences between the genotypes in two different cohorts of wild-type and NRF2 knockout mice that we could analyze. But when later we generated knockouts of NRF2 using a CRE driver whose expression is restricted to the thyroid and the kidney, there were no differences in T4 levels anymore. And all the other phenotypes I showed you were preserved. This means that this difference in taken together with the fact that TSH levels were the same, means that this is a difference in the total amount of hormone likely having something to do with its transport in the blood, which may be different in the knockouts compared to the wild-type mice. But this does not affect the levels of the functional hormone, the free thyroid hormone, the fractional is not bound to transport proteins, which is the one that exerts the metabolic actions and also regulates the levels of TSH secretion by the pituitary. So basically, no impact on thyroid function. So this was puzzling for us, and we think it may be because, on the one hand, NRF2 regulates positively the amount of thyroglobulin, but it seems that on the other hand, it regulates negatively the iodination of thyroglobulin. And here, it's the same gels and westerns I showed you before, but in addition, we have here the levels of iodinated thyroglobulin in single mouse thyroids. You can see that in the knockout mice without iodine, the levels are increased. And interestingly, when the wild-type mice are given iodine, the levels of iodinated thyroglobulin after 1 week are decreased, whereas the levels in the knockout mice are probably even higher than in the knockout controls. So the effect of NRF2 on thyroglobulin iodination seems to be opposite to its effect on total thyroglobulin levels. And we think that this is the way by which cells are able to use NRF2 to launch antioxidant responses when necessary in the thyroid without perturbing thyroid function. So this allows the cells to maintain normal levels of thyroid hormone production, we believe. We cannot model this for the time being mechanistically and investigate it mechanistically like we did with thyroglobulin production, because for this we need three-dimensional structures, we need follicles. So we cannot do this in two-dimensional cell cultures. And it seems that for some reason, so we have tried this with a protocol that exists to generate thyroid follicles starting from mouse epithelial stem cells. But NRF2 knockout mouse epithelial stem cells do not like to form follicles. Perhaps they are too sensitive to the protocol is too sensitive to the to oxidative stress that these cells are likely under. So for now, we have not been able to study this. We plan to approach this by using organoids from adult mice, wild type and NRF2 knockout mice. So to make organoids directly from the adult mice. So now I will talk about what happens when there is too much NRF2 activity. And there was a study from Japan describing a family.
Sorry, Jerry, you have a few more minutes in order to try to keep the time.
Okay.
Thank you.
Yes, thank you. In this family, there was a mutation in KEAP1, an inherited mutation that was segregated in an autosomal dominant manner. So these individuals who have the mutation here in black, they also had goiter, meaning large thyroids with multiple nodules inside. This is a mutation that was shown to activate NRF2. We studied the mice that have decreased expression of KEAP1, and we found that they had enlarged thyroids. So they recapitulated, at least in part, the human phenotype. They did not have nodules, but a diffuse enlargement of the thyroid. This goiter was already present in most mice by 3 months of age, and it increased further, became clearly 100% penetrant and more prominent in 1-year-old mice. And you can see here on histology that these mice, they had enlarged follicles. These mice, they had slightly increased, they had increased TSH levels when they were older. and this allowed them to maintain normal levels of thyroid hormones. So they had what we call subclinical hypothyroidism, a mild form of hypothyroidism. NRF2 was activated in the thyroids of these mice, as shown by the levels of the genes I showed you and the protein levels of NQ1. And thyroglobulin, interestingly, was not increased in the thyroids of these mice, but degraded. And we think this explains why they have this mild hypothyroidism. So these are degradation fragments of thyroglobulin in the thyroids of these mice. Since I'm already late, I will skip the rest. I talked to you about the antioxidant response, the involvement of NRF2 in iridotic metabolism, thyroglobulin expression and iodination, in goiter and hypothyroidism. I skipped the part on Hashimoto's thyroiditis and cancer. I think because the thyroid gland is the major determinant of basal metabolism, it would be interesting if you, or even necessary, I would say, if you are studying metabolism in relation to NRF2 signaling in vivo models, you should think about thyroid function. If you're studying metabolism in vivo models, you should think about thyroid function already. And if it has to do with NRF2, I think our work says that it should be addressed specifically. If you're working with animal models of genetically or pharmacologically altered NRF2 activity, it'd be interesting to look at the potential thyroidal effects. If you have samples from human trials of NRF2 modulated compounds, it'd be interesting to look if there are thyroidal effects. I didn't have time to show you two studies we have done where we looked in clinical trials, and there was no impact of either sulforaphan or dimethyl fumarate in thyroidal parameters in those clinical contexts. But this is the safety for the thyroid of NRF2 activators, I think, is something that should be addressed for each drug specifically. Finally, we need more families to understand goiter in humans and its relation with NRF2. So if you have access to human cohorts with germline mutations in the NRF2 pathway, it is definitely important to look at their thyroids. And with that, I will end here and thank my research team and collaborators and funders, and again, the organizers and the vendor for action for the invitation and the opportunity to present. Thank you, and sorry for going over.
Many thanks, Jerry, for the very interesting talk, and I give back the floor to Antonio to introduce the third speaker.
Okay, thank you very much. I think it's time to go to the next seminar, the next lecture. It will be presented by Dr. Nesrin Kartsal-Otser. She is a professor at the Marmara University, Faculty of Medicine, Department of Biochemistry in Istanbul, Turkey. She graduated in pharmacy from the Hacettepe University in Ankara and received her PhD in biochemistry in the same university before moving to Marmara University in Istanbul. She was a visiting scientist in prestigious institutions of the UK, Switzerland, and Germany, and has published over 100 articles and has been invited to participate in around 80 international meetings. She was the organizer of about 10 scientific meetings and conferences. As a member of international organizations, she has an impressive record of recognitions. For instance, as a member in the International Advisory Committee of the Oxygen Club of California from 1999 to today, in the FEBS Award Advanced Course Committee member in 2003 to 2006, the FEBS Fellowship Committee member in 2021, still going to 2024, the General Secretary of the European Society for Free Radical Research, and also President of this society in 2013-2014. She was awarded the Oxygen Club of California and the Lifetime Honorary Member in 2006, and also the Lifetime Achievement Award of the European Society for Free Radical Research in 2019. Her research is focused on endoplasmic reticulum stress redox signaling, and cell death in metabolism-related diseases such as cardiovascular disease and NASH. Thank you very much, Nerrin, for accepting this invitation to speak here, and the floor is yours.
Thank you very much. Thank you very much for the introduction, and thank you very much also for the invitation to speak about research. Cholesterol, as you all know, is a very important molecule, and it is found in all the cells of our body. And it is very important. It has structural importance. It is found in the cell membrane. It is important for synthesis of steroid hormones. But if its level is high, it might cause some metabolic diseases, neurodegenerative diseases, cancer, renal diseases, obesity, aging, diabetes, atherosclerosis, and non-alcoholic fatty liver disease or NASH. Here, non-alcoholic steatohepatitis represents a global health concern in all the countries, everywhere. It is characterized by fatty liver, hepatocyte cell death, and inflammation, which are associated with lipotoxicity, endoplasmic reticulum stress, mitochondrial dysfunction, iron overload, and oxidative stress. As you see here, progression starting from a healthy liver, first the lipids accumulate, fat accumulation in the liver, and it is called steatosis. And in the further stages, inflammation plus tissue scarring and fibrosis is added to this stage, and then it's called NASH. And in the further stages of this liver diseases, scar tissue replaces the liver cells, and cirrhosis, and even further stages, hepatocellular carcinoma. Cholesterol is important for the progression of NASH. Besides the free fatty acids, hepatic free cholesterol is suggested as an important molecule in NASH development by inducing inflammatory pathways in hepatocytes and enhancing fibrosis in hepatic stellate cells. There are many studies showing us this point. And also, epidemiological studies provides supporting evidence for an association between dietary cholesterol intake and NASH progression. Okay, and today I don't think I need to introduce NRF2 because we are in cost action, which focuses in pharmacological regulation of NRF2, and today's seminar is focused on NRF2 and metabolic diseases. NRF2, as we all know, is a transcription factor and is a master regulator of multiple cytoprotective responses and key molecular link among many many metabolic diseases. And it provides a unique strategy for drug development and repurposing for pharmacological and clinical area. This slide shows us relation between NRF2 and non-alcoholic fatty liver diseases. During the progression of NASH, as we know, from normal liver to non-alcoholic fatty liver disease, in this process there is a lipid accumulation. Fatty acids, triglycerides, they accumulate in the liver. NRF2 is important in this step because it regulates fatty acid metabolism and it inhibits de novo synthesis of fatty acids, and then it regulates this accumulation. And NRF2 also regulates or eliminates reactive oxygen species and increases antioxidant defense, increases expression of antioxidant enzymes, and then regulates oxidative stress, inhibits oxidative stress. By these two important mechanisms, NRF2 has important role for NASH development. There are studies focusing on NRF2 and NASH, and very recent publication in FRBM from the group of Jan Hayes, very nice review. They mention about relation NASH stimulating diet, for example, high-fat diet, atherogenic diet, or high-carbohydrate diet, induces steatosis in liver cells, and this steatosis induces endoplasmic reticulum stress and causes oxidative stress, and this causes inflammation and finally NASH formation. And also they mention NRF2 knockout animals, a knockout situation. Under these conditions, mitochondrial function will decrease, and some antioxidant enzymes, expression of some antioxidant enzymes will decrease, and will cause increased oxidative stress, inflammation, and NASH. In the same review, I like this slide very much because I think this slide is very good for our scope for this action, showing us NRF2 activity during NASH progression. At the very early stage of the disease, diet stimulated oxidative stress causing activation of NRF2, as we see here. But in the mid stage of the disease, which is here, persistent oxidative stress suppressing NRF2 and including this signaling pathway, and also in the late stage of NASH, prolonged oxidative stress and cytokine production causing robust downregulation of NRF2. This is important to see the activity, NRF2 activity in different stages of this disease, because a pharmaceutical that restore homeostatic NRF2 activity is very important. For example, for the late stage, we need to inhibit KEAP1 and TGF signaling, but for the mid stage, we need inhibit KEAP1, and for the early stage to inhibit KEAP1. This is very important for this. And another review also published this year in Frontiers Cell Development Biology, and they also emphasize the role of NRF2 in liver cancer. Here they mention about liver cancer risk, and reasonable NRF2 levels may inhibit tumor, but abnormal stimulation of NRF2 can promote tumor progression. Therefore, dose of NRF2 drugs should be carefully determined, and pharmacological activation of NRF2 for therapy should not exceed the therapeutic window. This therapeutic window is very important. Okay. Another study, again recently published in FASEP, also very nice results. In this study, they study with individuals who has a non-alcoholic fatty liver disease and compare their NRF2 level in the liver with obese mice liver data. And as we see here, in the patients with non-alcoholic fatty liver disease, they have increased expression of NRF2, and also in the obese mice, they found the same result. They did very detailed studies, and this study mentioned it is urgent to further study the role of NRF2 for NASH, and they found hepatic NRF2 expression level was upregulated in patients with non-alcoholic fatty liver disease and obese mice. NRF2 deficiency exhibited antioxidant, inhibited antioxidant response and promoted chronic inflammatory response and macrophage infiltration in the liver. NRF2 deficiency was observed to promote development of hepatic lipid deposition when mice were fed a high-fat diet. SREV1C activation was identified to be crucial for NRF2 deficiency to promote fatty acid synthesis. NRF2 deficiency was found to inhibit lipolysis by weakening autophagy. Today, I'm going to, after this introduction, I'm going to mention about our results because our research focused on recently to determine effect of cholesterol in non-alcoholic fatty liver disease and NASH transition in a manner of lipid droplet biogenesis by using in vitro and in vivo models. As in vitro model, we use mouse hepatocyte cell line, I will mention later, and loaded cholesterol. And as an in vivo experimental model, we use the rabbits fed high cholesterol diet. And for many years, we focused on the role of alpha tocopherol by using in vitro cell culture studies and in vivo studies, experimental atherosclerosis model and NASH models. Therefore, I will also mention about role of alpha tocopherol, a protective role of alpha tocopherol. As an animal model, we took male albino rabbits and we took them in 4 different groups. First group received only the diet, second group received 2% cholesterol in diet, and the third group received 2% cholesterol in diet and also alpha tocopherol intramuscular injections, 50 milligram per kilogram, and alpha tocopherol as a control group again. And after 8 weeks later, serum samples and liver tissues were collected. And here we established lipid accumulation in the liver tissues. And before doing that, we measured serum cholesterol level of these animals, as you see here, in the control group, and in cholesterol received group has very high, significantly high cholesterol. They are hypercholesterolemic. And the group who received high cholesterol plus alpha tocopherol, again high cholesterol. And we measured their serum triglyceride levels. Group who received cholesterol high triglyceride level, who received alpha tocopherol, again a little bit high. And we also checked serum alpha tocopherol levels, and animals who received alpha tocopherol has high alpha tocopherol levels in their serum. And then we look their liver macroscopically, as you see here, control from the control group, normal appearance of liver. And the group who received cholesterol, their cholesterol level is very high in their serum. And as you see here, yellowish liver macroscopically. And this group received cholesterol plus alpha tocopherol, again yellowish. And this group received only alpha tocopherol. And when we take their histological sections and look them by oil red, and here you see the red spots shows lipid accumulation in the liver of these animals. And alpha tocopherol received group has less lipid accumulation here. And also we evaluated liver triglyceride and cholesterol level of these animals, and in the liver cholesterol and triglyceride also accumulate. In a way, we checked by giving high cholesterol in the diet, these animals get non-alcoholic fatty liver or NASH progression. Then we checked we checked the livers of these animals by immunohistochemistry, different parameters, as you see here, RAM11, which is a marker for macrophages, macrophage accumulation. Here you see in cholesterol treated group, you see macrophage accumulation and inflammation. And also alpha tocopherol received animals has less macrophage accumulation. And alpha SMA, this is another marker and for fibrosis, and cholesterol received group, as you see here, fibrosis formation, and less fibrosis, alpha tocopherol treatment, and this is control. And another one is called 1A, collagen accumulation, accumulation in liver tissue. In cholesterol treatment, as you see here, more collagen accumulation, fibrosis, and alpha tocopherol treatment decreases this situation. Then we looked oxidative stress markers for lipid peroxidation, HNE. We checked HNE by HNE, and in cholesterol treated animals, you see lipid peroxidation, HNE, we found more, and alpha tocopherol treatment, as you see here, less HNE and less lipid peroxidation. And we checked the 7-ketocholesterol amount in the tissue. Why we did this? Because excess cholesterol gets oxidized, and oxysterol forms, and 7-ketocholesterol is one of the oxysterol, and its level increased in cholesterol treated animals, and its level is less than in this group because they received alpha tocopherol. All these results from the liver of these animals. Then we checked NRF2 in the liver of these rabbits, and the group who received cholesterol, and we, as we see here, NRF2 expression increases significantly. This animal has NASH progression. How do we know? Because we know from microscopic data also. And alpha tocopherol treatment decreased NRF2 expression, as you see here. In a parallel, we looked at cypin expression. Why cypin? I will explain you later. I will give you later about cypin, because cypin is a protein which is related with formation of lipid droplets. Therefore, we evaluated cypin level in this liver, and cholesterol treated group also cypin expression increases, and alpha tocopherol received group has less cypin expression. Okay. We publish this data, and we have more data in biofactors, you see here. And if we put all the data we got from in vivo studies, we can say high cholesterol diet induces NASH in rabbits. And we checked oxidative stress markers. High cholesterol diet increases oxidative stress because increases NHNE, 7 ketocholesterol, NRF2, and alpha tocopherol treatment reduces oxidative stress markers. High cholesterol diet increases inflammation because we found TGF-beta goes down, interleukin-1 beta, RAM11, phospho-C-Jun, GNK all goes up, and this is reduced by alpha tocopherol treatment. And we looked autophagy markers, P62, Beclin-1 goes up by high cholesterol and goes down by alpha tocopherol treatment. And apoptosis markers, caspase-9, CHOP, BAX goes up, increases by high cholesterol diet, and CHOP and BAX goes down by alpha tocopherol treatment. And fibrosis markers, alpha SMA, goes up by high cholesterol diet, and alpha tocopherol reduces fibrosis in these animals. Okay, and by using the same animals which we use for NASH model, we also checked their aorta, and we checked their progression of atherosclerosis, and we found atherosclerotic plaques, and we published these results in this paper. Why I tell this? Because in the same animals, aortic tissue, we found endoplasmic reticulum stress markers. Also, they are all ER stress markers increased in the aorta of high cholesterol treated group, and alpha tocopherol treatment decreased endoplasmic reticulum markers. And moreover, high cholesterol diet affects expression of proteins involved in redox hemostasis, and NOx also increased in cholesterol-treated animal, and NRF2 also increased in cholesterol-treated animals in their aorta, not only in their liver, but also in the aorta. And alpha tocopherol treatment decreased NRF2 level. So after we get in vitro results from NASH model, then we wanted to see or to check some parameters in order to understand molecular mechanism in vitro model, in cholesterol accumulated hepatocyte model. We used AM12 cell line, and we make them cholesterol enriched by liposome. Okay, and we took these cells, and then by using liposomes, and here only liposome, and here liposome loaded with cholesterol in different time intervals, starting from 0 control up to 10 hours, we check cholesterol accumulation here. And what we have seen, cholesterol accumulates, especially after 6, 8, and 10 hours, it goes to maximum level. And then we use 8 hours incubation to load cholesterol. And then we checked total number of lipid particles in each cell. And this one is liposome loaded cholesterol. And then after 8 hours, which is the maximum enough level, and then we evaluated total number of lipid droplet per cell. There is an increase here, but the difference between these hours were not significant. Then we checked different diameter lipid droplets, because lipid droplet size alters. There are two types: small lipid droplets, their diameter of less than 3 micrometers, and the large ones larger than 3 micrometers. So we checked, we evaluated regarding their size. As you see here, small lipid droplets, there is no significant change. They are also increasing, but there is no significant change. But when we looked large lipid droplets, as you see here, at the 8th hour, there is a significant increase in large lipid droplets. Then we talked, what is the relation between lipid droplet biogenesis in cholesterol accumulated hepatocytes? If there is any relation between cypin and also NRF2. What is cypin? I mentioned before, cypin is an endoplasmic reticulum anchored protein, and which is mostly studied, extensively studied in lipid droplet biogenesis. As you see here, this is an endoplasmic reticulum, membrane endoplasmic reticulum, and this purple protein is cypin. And it is important for the progression or for the formation of lipid droplets. This one, for example, nascent lipid droplet, now it is forming, and this one is mature lipid droplet, and cypin is very important for their formation. Under the conditions cypin knockout, as you see.
Sorry, Nazrin, we need to try to reach the end in order to leave some time for discussion.
Okay, I have a few slides, not very much.
Yes, thanks.
And then for the maturation, we need cypin. If cypin is knocked out, then no maturation for lipid droplets, then lipids will not accumulate, of course. Then we set this system for efficient sRNA delivery and optimizing of cypin gene silencing. And at the 48 hours, cypin silenced. Okay, I will skip this. Then we evaluated NRF2 HO signaling. Therefore, we evaluated NRF2 and HO1 mRNA levels. Here, when we silence here cypin, there is not a big difference for NRF2. But when we silence cypin, before silencing cypin, HO1 goes down in cholesterol loaded situation. But if we silence cypin, then HO1 goes up in a way reverse to first situation. This is very preliminary data, but we are working, focused and working on it. I hope next time I will have more data for this subject. In conclusion, from our in vivo studies, we can say lipid droplet biogenesis together with cypin levels and NRF2 signaling are increased in liver tissues of rabbits. Alpha-tocopherol has the ability in inhibiting high cholesterol mediated non-alcoholic fatty liver disease and NASH transition, which might be related with reduced inflammation and oxidative stress levels, possible effect of NRF2. From our cell culture studies, inhibition of lipid droplet biogenesis via cypin silencing reverses HO1 reduction. Our findings show the possible relation between cypin and NRF2 levels that is regulated by cholesterol-mediated lipid droplet biogenesis in NASH. Finally, I would like to thank to the people who is working in my group currently, and the grants from my university, Turkish Research Fund, and some cost actions which I was involved, and my collaborators, Azzi for alpha-tocopherol studies, Gurune, Giovanni Man for NRF2 studies, Teybas, Özcan, Galli. Thank you for your attention.
Thank you very much, Nazreen. Very, very interesting. So I ask now all the panelists to come back to the video, also, of course, Antonio Quadrado. And we open the floor for discussion. Already there were some questions in the chat. Maybe, Alke, would you like to elaborate a bit more about the different questions you received from Giovanni Mann, from Amedeo Columbano, and Amy Egler? If you want to add anything, please do so.
Yes, sorry, I just dropped off my video, so here I am again. So yeah, concerning the Giovanni's questions concerning the AMPK's insensitivity related to the insensitivity of GCLC to the different oxygen concentrations, this is definitely a very interesting point we need to look into, so that we are not dealing with some sort of artifact in our system which nothing has to do with some physiological context. So I think this is definitely an interesting point to look at, whether these AMPK independent genes are actually the ones which are not really responsive to the ambient or to the environmental oxygen concentration. I think this would be really interesting to look into. And I think there was another question from Amy, whether we tested other phytochemicals, whether we do see the same effect in terms of AMPK dependency on the investigated genes. And indeed, we checked a couple of, and there was indeed the same thing. But as I said, there was no systematic investigation whether it's a type how NRF2 is activated and whether there's accompanying AMPK activation by the same compound. So I think this would be really a cool thing to investigate, especially with natural compounds, because they are so polypharmacological. So I think like dual activators of NRF2 and AMPK signaling might have maybe synergistic effects on the cellular stress resistance. So this would be super cool to investigate. And I think there was another question in terms of how P62 is phosphorylated and how AMPK can be activated. So P62 phosphorylation can be also mediated by other kinases.
From
that's for
Medeo
sure.
Columbano, he was asking about phosphorylation of P62. You replied already in the chat.
Okay, good. All right.
Yeah.
So then it's going to be fine. So I think these were the questions, right? So if there's any, just remind me. I'm happy to answer, but I don't see any more questions.
Yes.
All right. Oh, we lost Luciano.
Okay, I think
yes.
Luciano is coming back in a minute. In the meantime, maybe I can have a couple of questions for Nesrin.
Yes.
You know, we are having many different models for NASH, and you are having wonderful data with tocopherol in these rabbits. But I guess I wonder how reproducible this is in other models, like the one with fructose, the one with F5, the STAM model. So what do you think about the reliability of these models in humans, and whether you think that your data will be possible to extrapolate to human beings?
I think there, as you said, there are other models such as high fructose diet. We also planned to do by using different animal models. We applied for project. So since we didn't, I don't know the results. But for rabbit studies, we did previously, we ran, and then now we are using some mice models again, but we don't have the data yet. But maybe next year in this action, I may present some results from knockout mice models.
Thank you. Are there more questions? Maybe some question for Jerry. So I was surprised, Jerry, by your data showing that these people overexpressing a bit of NRF2 because of the KEAP1 knockdown or hypomorphic form of KEAP1. And the next day results that you got also in the mice. So how often, how frequent is this kind of mutation in the human population?
These mutations are rare. This is, for now, there is only this one family plus one individual. This family is a convincing case because it was a clear loss of function mutation, and there was perfect co-segregation over several generations of the mutation and the phenotype, the goiter. There is also one more description of a single individual with a missense mutation and goiter. What is also interesting is that both of these were described in Japan, where iodine intake is high, basically due to the consumption of algae and products from algae. So, and also in our mice, we have some evidence that suggests that the phenotypes we observe in these mice, they depend on the iodine intake. And we're working to characterize this. So this may be an example of the genotype-environment interaction. To come back to your question, these are very rare families or individuals. We are working to do a reverse genetic approach, meaning because goiter is very common, but KEAP1 mutations are very rare, inherited mutations. So we are now looking at cohorts where we have mutations to characterize the thyroidal phenotype. And what is very striking is that what brought this particular family to medical attention was the goiter phenotype. But KEAP1 and NRF2 are expressed everywhere in the body. But what gave a phenotype, brought them to medical attention, was the thyroidal effect. Maybe it's
Very
because the
interesting.
thyroid is in the neck and you can see it when it grows, or maybe it's the most sensitive organ to NRF2 manipulation.
Thank you very much. And Giovanni, I think he has a question.
Two questions, or three, if I may. Otherwise, you tell me to be quiet. One is for Elke. I have a question. Has anyone looked at AMP kinase? Because you said it phosphorylates P62, autophagic digestion of KEAP1, etc. In an P62 knockout mouse, what does NRF2 do? Well, what does AMP kinase do to NRF2 target genes?
That's interesting. So
Okay.
Actually, I have to admit that I don't know it by heart whether this has already been done, but I think it's definitely worthwhile to check into that, whether how the autophagy pathway is really acting on this, whether it's like the predominant one. I would assume that I think it's cell type dependent, likely. So I would assume that liver cells are really dependent on the P62 axis in terms of AMPK susceptibility for NRF2 activation, whereas others are rather going like brain or so, rather goes into the GSK3 beta. But but I think it's cell type dependent, but I'm not 100% sure, and I don't want to waste your time by speculation.
I should know, but I'll ask my colleague in Tsukuba University, Tetsuro Ishii, who was working with the P62 knockouts. My other question you may not be able to answer is, if we go in vivo and we have AMP kinase working in a blood vessel, for sake of argument, and we know that the blood circulating has a good oxygen, physiological normoxia, do you think that GCLC and GCLM, the key glutathione enzymes, are the ones that are responding to changes in oxidative stress locally? Because if we take things like NQ01 and HO1, and we do it in cell culture in hyperoxia, they're massively induced by sulforaphane dimethylfumarate, beautiful. But if we take them into physiological normoxia, those two particular genes are not activated dramatically. They still get activated. So in our paper some time ago, we speculated that the glutathione synthesizing genes are critical or may be critical in vivo. And I don't know enough about AMP kinase, what it does under in vivo conditions.
The thing is that AMPK itself, it's also dependent on the oxygen concentration. So I think it's also a redox sensitive protein. So I think it's pretty hard to predict, but I would assume, likely it wouldn't be such a traumatic thing in the blood, which is happening.
And the other thing I was going to do was just a comment. BAH1 protein levels are upregulated, believe it or not, in hypoxia. But hypoxia has a definition. Less than 1% oxygen. It doesn't mean just lowering oxygen. And it's also upregulated in hyperoxia, all the conditions you culture your cells in. But if you do your AMP experiment in your cell type, whatever it, I can't remember what it was, but if you did it in a cell that's adapted to 5% oxygen, BAH1 protein levels will be low.
Okay, so that would be really interesting to see.
So you could,
recapitulate the effects depending on the oxygen concentration.
Remind me of the cells that you used. What cells?
I mean, so far they were just fibroblasts, so I think they were pretty artificial anyway. Yeah, that's true. So actually, in the long run, we also wanted to go to some more physiological system, but it's definitely a good point to look into the oxygen concentration.
Cool. And then this is just wearing another hat, if I may, as the editor for reviews and special issues in FRBM. Is there much written in your field on AMP and NRF2 and autophagy?
For autophagy, I'm not sure. I think there are a couple of papers and they mainly end up with mTOR signaling in the end. But with the P62 axis, I mean, there are a couple of out there, but I think not particularly focused on this. There might be still room for improvement. Or
have you written recent reviews?
We are just writing one on AMPK and NRF2 for your special issue.
She has just been accepted one review article in
our issue.
Special issue.
Thank you.
Sure, welcome. Thank you for being in there.
Good. That was all.
So Luciano, would you like to go on?
Yes, thank you, Antonio. In the meantime, I see in the chat there are no other questions, so I encourage all participants to put more questions. We have a few minutes more for discussion. There were some positive comments by participants, which is good. Maybe a general question for Nasrin in relationship to the alpha tocopherol in the development of NASH. In terms of a clinical translation, what can we expect, from the results that you got?
Clinical studies also show epidemiological studies or retrospective studies show people who has high vitamin E levels in their blood, their NASH risk is lower. Mainly people who has very low vitamin E levels, they have NASH progression. And also this gives good signal alpha tocopherol might have beneficial effects for the NASH treatment. And also our results also show us.
Thank you.
So if I may have a question also for Nesrin. Yeah, we also saw this data indicating that somehow NRF2 downregulates fatty acid synthase. Can you provide some mechanistic insight into this?
NRF2 inhibits fatty acid synthesis because it regulates probably SREF1 expression.
But it's regulated by inhibiting SERBP1 expression. Does it inhibit the expression of this transcription factor? So this is the point. I think there is evidence that fatty acid synthase is downregulated. There is also evidence that beta oxidation of fatty acids is increased, but maybe the mechanistic link is still not well defined. And this is something that it would be good.
There are studies, as you said, there are studies showing contradictory results, and also many studies suggest we should go in detail. More research is needed in this respect.
Thank you.
Thank you.
I ask again if the participants have any other question. We do have a few more minutes, not so many. Or if there is anything else that also the panelists would like to add, maybe to their talks, based also on the discussion that we are having, also please feel free to intervene.
Giovanni, we have some extra time for your questions.
Yeah. I was going to ask if we have another, he's asking me, another question for Nesrin. In the last part of your talk, I didn't put it in the chat. You showed that is it called SEI-PRIN, that if you knock that out, you augment the signal for HO1. In other words, I didn't follow that, and I don't know if Antonio asked you the mechanism just now because I was looking away from the screen, so I apologize. But I hadn't seen that data before and found it interesting.
Yeah.
But
yes.
Did Antonio, did you ask that question already?
No, different.
Okay. No.
All right. So do you have an insight as to why that's happening? When you knock down that component, why suddenly the lipid-induced induction is higher?
Yeah, we don't know yet, because it's, as I told you, as I mentioned, it is our preliminary data, and we are working on that, and we are very curious. We have many questions in our mind, but if you have some suggestions.
Well, I must say I'm pleading ignorance because I didn't know much about it until you spoke about it. So I would have to look that up. The other thing, while Antonio gave me the floor, for all of you best friends of ours or mine, I'll be setting up in the end of October here in London a physiological oxygen workshop, but only restricted to pharma. So I'm inviting not pharma marketers, the distributors, but pharma researchers to educate, if I may use that term in inverted commas, the critical importance of doing high throughput screening using the Perkin-Elmer device, 1.2 million pounds under oxygen. It's not hypoxia that's critical, except if you're a cancer person. But I'm really, really seriously now convinced that if we want to understand redox signaling, efficacy of drugs, time-dependent, concentration dependence, we've got to encase that unit, which is a lot of money. And this is a big R&D development. So watch the space. If I have a program, I'll advertise it through Ben Farr, right? Bench to what, bed to bedside type thing, and let you know.
Send me a note, Giovanni. Send me a note, and we will put it in the next newsletter.
I will do that. And if you have any pharma companies that you're working with that you think are interested scientifically, but don't want to do it because it would possibly negate everything they've ever done. My point is, it's not about the past, it's the future. And if we want to redesign application of a drug, be it a statin, for example, we should have looked at these things in cells under this oxygen level. So anyway, watch the space. And the other thing I was going to say, I gave you information, Antonio, about Siena. I wrote you a brief thing with the program. That was one of the best meetings I have ever had the privilege of attending, to be an old timer sitting in the audience, listening to Ana Rojo and the others speak. It was unbelievable. And they were excellent. I don't think at that age I could have done the same. I didn't have the capacity. But these people spoke as if they were our age. And all the old fogies like me sat in the audience listening to well over 80 young people speaking. Amazing. So it's something we should consider for the future. Rather than have all the professors and everyone speak, let us educate the future of our youth. So I think if you talk to Anna, she would have been happy with the meeting. Okay. Anyway, I have no more questions. I'm sitting in lovely Lisbon. I'm going to have some little drink.
So maybe, Luciano, you want to talk about the next meeting? It could be probably end of September or beginning of October.
Yes, we are planning to continue. Then we will inform all the participants. So when you register in this series, also you get, in a way, in our loop, receive soon more information on this series. It's a pleasure to collaborate with Antonio on this, and also let me thank again all the other, organizers from Italy. Mauro Maccarone, Alessandro Giufflé, Marzia Arese, Elena Forte, and Alessandro Leuti. So we are a big team. So everyone is contributing, and thank you to everyone. So to conclude, maybe Antonio, if you agree, we can ask maybe the three panelists to give a final statement,
yes,
if they wish to do so, and then we should close at 5 p.m. I think many of us have to jump in another Zoom probably. Why not? I invite you that you will have a little drink, because we are jumping in another Zoom here. So thank you. Please, Elke, if you want to start.
Yeah, I think actually the title of this webinar brings it quite nicely to the point which I would like to make. It's that NRF2 and metabolism shouldn't be separated. So I think they shouldn't be seen as distinct entities in the cell, but they're really like influencing each other. So, and it's not a one-way direction. It's also that redox influences metabolism and metabolism influences redox homeostasis. And I think to understand this relationship would be really interesting and very important to deal with all these diseases which are caused by stress, be it redox or nutrient stress. So I think this is really a way to go, to look into this interplay. Yeah, that's from my side.
Thank you very much, Elke. So, Jadi, would you like to continue?
Yes. My message is simple. If you're working on metabolism, or if you're working on NRF2, or if you're working on NRF2 and metabolism especially, don't forget about the thyroid.
Thank you. Very, very clear message. Nasrin, please.
Yes, I would like to say this action is very good topic, has very good topic, and congratulations, Antonio and co-workers, because the data about NRF2 and the diseases and other conditions not very clear, really needs many studies. And many reviews, recent reviews also emphasize this. The data, there are contradictory data, and more research is needed to clarify NRF2 and, let's say, metabolic diseases.
Thank you, Nasrin. So from my side, I would like to conclude. Also thanking very much Brigitta Buttari, that is working very hard with Antonio and with me for this series. Many thanks to you, Antonio, and I leave you the floor for the final word.
I just want to say really thank you very much to both to the speakers, to the organizers, and also, of course, to the 57 people who still remain in the room. And what I really like from this metabolic approach to NRF2 is to distinguish, if possible, just leave you with the idea of distinguishing between redox signaling and oxidative stress. And I think that most of the work that we are doing in metabolism has to do with redox signaling rather than with oxidative stress. But this is something to discuss in another occasion. Again, thank you very much to all of you.
Perfect. Thank you. Thank you very much to all of you.
Thank you.
Thank
Bye-bye.
you. Thank you for your attention, and thanks for having me. Bye-bye. Take care, everyone.
Bye.
Bye.